We asked Ian Affleck, Vice President of Plant Biotechnology at CropLife Canada. What is the evolution of plant breeding?
So as we talk about plant breeders and the evolution of the activity of plant breeding, you can really trace its roots right back to the beginning of agriculture almost 10,000 years ago. So as hunter-gatherers started realizing that it's a lot less work if I just drop seeds outside my hut and grow the wheat there than it is to go chase it in a field somewhere, hunting and gathering wheat plants, if you will, then we became agriculturalists. And once we started agriculture, we started to realize that if we just kept the best seeds, the most hardy or the ones we like the most, that the next year they would resemble those more. So those really early agricultural ancestors were selecting for genetics. They just didn't know it. They didn't realize they were selecting for certain genes, but that's what they were doing. They were picking the plants that worked best for them in agriculture and continuing them forward. And then as that kept moving on, we domesticated more crops. We started growing them in rows and really started to look like the agriculture that we see today. But that really was the limit of technological advancement for about 7,000 years. Then we really started to notice that if we really picked the plants that expressed the plant parts we like to eat the most, we could just have more of those plant parts. So a good example is broccoli in the form that we see it in today really didn't exist before about 600 BCE. That's when we started taking wild mustard and just choosing the pieces we like to eat the most, so they got bigger and bigger. And that wild mustard or wild cabbage plant basically turned into broccoli. And I'll talk a little bit more about that because it's one of my favorite examples of kind of how we selected crops to work in agriculture the way we needed them to. But after that, wasn't really a lot of advancement until the 1700s, where we started to realize that you can cross different plants that are closely related. So the example in the little picture here is a grapefruit, and grapefruit didn't exist before 1750. Someone planted a Jamaican sweet orange and an Indonesian pomelo beside each other on a tropical island, and they cross-pollinated, and then they got this really tasty fruit that came from it. And that's how we ended up, through crossbreeding these two species, to get grapefruit. So it's another food we often think was around forever, but it's actually, in the history of agriculture, it's pretty new. So again, not a whole lot of advancement in plant breeding other than realizing we can do crosses until about 1863. Then, if you remember from high school biology class, we talked about Gregor Mendel and the smooth peas and the wrinkly peas and the heritability of genetics, kind of like the heritability of eye color when you think about it in humans. And he realized there's math behind this. There's a ratio at which these traits move. We can track it, we can understand it. So he really started to begin the understanding of genetics and how that all works. So that was a big leap forward in plant breeding, is knowing exactly mathematically how to move the genes we want from one place to another. So that was really important. Then you move forward to about 1940. And although we're moving genes we've wanted to over these years, there's something else we were doing that was really important, is like in a human, when you're not just the child of your mother and your father, you're 49% the DNA of one and 49% the DNA of another. And then there's a little bit of random mutations, uniqueness in you. And that's the same in crop plants. So when you make your crosses, or the next generation of the plant comes, there'll be little differences. And sometimes these can be really beneficial. So in the 40s, realizing that these rare and small changes are really beneficial, and that's what we're looking for in plant breeding, we learned ways to encourage those changes to happen more often, so that we could get to the changes we knew would arrive, but we could get to them more efficiently and deliver those better varieties a little faster. So there's a whole bunch of plant breeding techniques we started using in the 1940s to kind of drive that forward. Then you move on to 73, and a grad student comes up with essentially what we call rDNA, or that's what you'll hear in pop culture as GMOs, that invented that technique. And then that took forward to kind of bringing forward the first wave of genetically modified crops, or GMOs, with the first being grown in Canada to be corn, soybean, and canola. We still grow a lot of those today. Over 90% of those three crops are GM varieties here in Canada today. So from there, we move forward. Another kind of important milestone in the history there was the papaya industry in Hawaii was being devastated by a ring spot virus, and a university professor there endeavored to use that rDNA technology, that genetic modification technology, to make the plants resistant to that disease. He was successful. He gave that technology to the farmers, saving the papaya industry. in Hawaii. And now, 80-90% of all the papaya in the world is genetically modified because it's so successful in preventing that kind of disease. So moving forward to the 2000s, when we talked about, gene editing's one tool in a bucket of many, a lot of new tools came along to help that plant breeder do what they're doing. And that's genetic selection, it's marker assisted breeding. There's a whole bunch of things. They rarely get talked about. It's really gene editing and GMOs that are the ones that make the headlines, but there's a lot of different tools that are available to plant breeders, and they use them all in trying to bring those varieties to market. So then some other interesting milestones. In 2014, we start to see kind of a new wave of GM crops come through that are more consumer focused, that provide different traits like non-browning and non-bruising. So you lose less of the apple, whether you're processing it into slices or whether you drop your apple on the floor. This will help it not bruise as quickly, so you don't have as much food waste there. It's an easier food to consume. And similar for the potato, traits like that start coming along that help out in the food side of the equation. So this is kind of the history of where plant breeding has come from in the last 10,000 years. But just to kind of drive it home visually, some good examples here of kind of what wheat looked like 10,000 years ago. Tiny seeds, they'd fall off the head really easily. So if you're farming it, that's really inefficient because you're losing the seeds on the ground. So they selected them for bigger, stronger, better seeds until you get to the common wheat where the grain looks like it is today. Corn's another great example, which started as teosinte, which is the grass on the left. Little tiny plant with really hard seeds. You basically have to smash them with a rock into a powder so that you can make the flour out of them. And then through thousands of years of selection of choosing larger, softer kernels, you get to the modern corn that we have today, which is much better suited for agriculture and much easier for us to consume. Another great example is kind of what the wild banana looked like back then toward what it looks like now. By selecting for the genes that expressed smaller seeds and softer seeds, you got rid of those big seeds you see in that banana on the left that were inedible and got them down to the tiny little edible seeds that you see in the bananas we have today. So a lot more edible fruit in the same size banana. So that's kind of how we can really have that impact over thousands of years of selecting for the gene expression that we're looking for. That's what plant breeding is all about. And this is one of my favorite examples. If you take Brassica oleracea, which is wild cabbage or sometimes called wild mustard, depending on where you are or what part of the world you're in, you are selecting for different things you like to eat from that plant. So if you like terminal buds, you just select for those. And eventually it started to look like cabbage. If you just like the stem, it would start to look like kohlrabi. So all of these plants, Brussels sprouts, kale, broccoli, cauliflower, they're the same plant. They're all wild cabbage, just expressing different elements of their genes more than their counterparts. So these are similar to, you see a yellow potato and a red potato and a russet potato. Those are all potatoes. They're all the same. They just express different traits. That's the same here. These are all the same plant. They just express different traits. So I don't know who spent thousands of years choosing for Brussels sprouts. I wouldn't have spent time on that one personally, but that's all the same effort to get to food products that fit either the lifestyle or the culture or the region in which you live.
So what is gene editing?
So to talk about how gene editing works, it's important to reflect on how DNA functions in general. So DNA is like a long book of information that instructs how a plant grows. And scientists can now identify the exact page in that book, the exact paragraph, even the exact word they want to change. Then they can use gene editing tools to make that very specific improvement to the plant. So if you were to compare this to conventional breeding, and I'm oversimplifying, but if you were to take 2 pages of a book and you know you want one letter from this page to move on to the other page, but you want the other page to say entirely the same other than that one letter, conventional breeding is kind of like rubbing the 2 pages together until that word, that one and only one letter transfers. Now it's much more complicated than that, but that's essentially what it takes. And it takes plant breeders years and years and years and thousands of plant crosses to achieve that outcome that they know from Mendel's work is possible. They know they can do it. They just got to get it to happen. So gene editing allows them to go in and make that tweak very specifically to the gene that they know they're trying to influence. So a very effective tool. Now, and it's not only useful for plant breeding, it's also useful in human health, industrial applications, and animal health. But today we're really going to focus on the plant side of it. So when we say, so what is gene editing? We're talking about it goes in and makes specific changes, but what really is it? So I really enjoyed this graphic from a recent meeting. The government of Japan used this as they described gene editing during a recent OECD meeting, an international forum on the science of plant breeding and food. So it's a technology that allows scientists to make precise targeted changes to the DNA. And as the Japanese government has put forward, this smiley little CRISPR-Cas9, where we see all the colors lining up, that's because the Cas9, the CRISPR, the gene editing tool is designed to match exactly with the DNA that you're looking for, and then make the cut at the specific area that you're looking to make in order to affect the change that you would have been doing with your conventional breeding activity anyway. So the idea here is to be very focused, very specific, make the cut you need, and as the DNA repairs itself, it'll repair itself in the approach that you're looking for. So a really useful tool in plant breeding to get to those changes in a much more efficient manner. It's important to note two kind of revolutionaries in the space. This is Emmanuel Charpentier and Jennifer Doudna, the two scientists who discovered CRISPR. They saw that this was happening in nature and brought it out to use it as a plant breeding tool. And for any trivia night folks who want to know what CRISPR stands for, it's clustered regularly interspaced short palindromic repeats. I'm sure that will not stick in your head immediately after this, but maybe on some trivia night somewhere that might come in handy. But they're really trailblazers in their field and brought forward a Nobel Prize winning scientific method that's going to have great opportunities in plant agriculture and, as I mentioned, in other fields as well.
What is the difference between gene editing and GMOs?
Plant breeding. So this large bubble on the outside is plant breeding, with dozens of tools that plant breeders use to bring better varieties forward. But we're not going to talk about all of them, but I think it's important to compare what gene editing is and what genetic engineering is, which results in the kind of, if I'm using a pop culture term, GMOs that you'll hear talked about often. So the focus of gene editing is to work within the existing genetic code of the plant and make improvements that could have occurred in nature over time or through traditional breeding over time, but may have taken you a long time. Genetic engineering is adding new DNA from another organism, and those improvements are probably unlikely to occur in nature or through traditional breeding over time. So if we go back to the book analogy, gene editing is working on the page you know the book really well, and you want to make this one specific change. Genetic engineering is kind of bringing a page in from another book, a really good page that's going to improve the story, but it's a whole different page from a different book. So that's kind of the difference there. And they do overlap a little in the middle. And I think that's important to understand, because while we explain gene editing as focusing on the genes that are within the plant, it is possible to bring in DNA from another organism using gene editing. But if you were to do that, if you were to live in that crossover space, you'd be considered a GMO and you would be treated as such from a global regulatory sense. So really, when we talk about gene editing, we're talking about the parts of that circle that are not overlapping with GMOs, working within the plant's own genetic code to do what plant breeders were trying to do over time through traditional plant breeding. That's really what we're focused on. And it's important in that context. So when we understand the difference, what about the safety? So everything in that light shaded bubble, plant breeding, has been safe. Plant breeding is an inherently safe activity. It's resulted in thousands and thousands of safe varieties, including ones that have used gene editing and including ones that have used genetic engineering, which again are just two of many, many tools.
Is gene editing safe?
So as we talk about safety specifically, there is many scientific bodies around the world that have come out with statements describing gene editing and its associated safety. So gene editing is safe for use in plant breeding. A great summary paper was recently released by the Federation of Academies of Sciences and Humanities in the European Union, which stated very clearly that using gene editing to accomplish what you would have done in conventional breeding is equally as safe. And there's been a number of other associations around the world that have also come forward with similar statements, whether it be Agricultural Biotechnology Council of Australia, Food Standards Australia and New Zealand, a number of other European science associations. So gene editing is really just a more precise way to conduct your breeding activities. So it brings forward all the safety that breeding has had with it in the past. So when we talk about safety, And we mentioned that if you were in that crossover space where you would have created a GMO, what's the global landscape? People want to understand how these things are regulated. And we've seen from a number of countries around the world, like Australia, Japan, New Zealand, the USA, South American countries like Argentina and Brazil, have put forward declarations and statements that if you could have arrived at your endpoint with conventional breeding, then it will be treated as such. So gene editing focused on working within the plant's own genetic code won't be treated any differently than conventional breeding because it is inherently a more precise version of that same activity. So that's the global trend that we're seeing. There is some differences. Europe is currently having a strong discussion about how they're going to handle it. They have a legal opinion that it should be underneath their GMO regulations. Their science, as we mentioned before, their scientific academies are saying that's not the correct approach, and they'll be embarking on a discussion over the next year to decide how they're going to handle this in the EU. And there's a new report due to the Commission, I believe, in April that's going to really dive into that. So we're interested to see where they go, but from the majority of countries, we're seeing a similar pathway that if you're conducting what's essentially conventional breeding and incorporating gene editing, it will be treated that way with all the safety that comes with it. From a Canadian regulatory environment, we're expecting the Canadian government to begin consulting on how it's going to clarify its existing novelty rules and policies around gene editing. So we're looking forward to that public consultation to start any day now. And we're really hopeful that Canada will follow that global regulatory trend of recognizing the inherent safety of plant breeding, especially when it comes to using precise tools like gene editing.
What are examples of gene editing?
So a couple of fun examples. One is coffee. We've had some really serious disease problems in coffee over the last number of years. 2012 specifically was a real highlight year where 55% of coffee grown in Central and South America were lost to coffee rust. Just everything in that time just circled perfectly to make the right conditions for coffee rust to kind of wipe out a lot of coffee plants. A lot of people lost their jobs because of that. But plant breeders using gene editing in public institutions in South America are looking how to use gene editing to solve that. And they're making some really great progress in making coffee more resistant to that rust disease. So a great example of how it can be beneficial in that quest for disease resistance. I think this is a good example where often we think of GMOs, we think of gene editing as kind of delivering really interesting single traits like high fiber wheat or 100% rust resistant coffee plants. But where gene editing is really going to come in is that increasing the rust resistance by 10% with every new variety you put out. So it's not just these big blockbuster interesting traits. It's about helping plant breeders slowly overcome the problems that they're constantly having in agriculture. And I think that's one of the really interesting and often overshadowed parts. It's not all big headline news traits. It's that lifelong dedication of a plant breeder to bettering plants, and they're able to incrementally better them using this technology in their breeding programs. So another story is bananas, and you may have heard of this, that the banana that we buy in the grocery stores, the Cavendish banana, is one variety, and it's all clonally propagated or it's from cutting. So this is like potatoes. You don't grow potato seed off of the top of the plant and replant it. You cut a potato in half and you put it back in the ground. So it's the exact same potato every year. So that creates a susceptibility to potential diseases. So because you can't crossbreed with another banana without losing a lot of the benefits of the Cavendish banana, gene editing would allow you to go in and solve some of these disease issues, a lot like the papaya issues we had in Hawaii, in order to manage a number of these diseases. So some might say, well, why not go back to the wild banana, which has more disease resistance? And you could, but you would go back to a plant that yields a lot less. You'd need more banana groves to grow the same amount of banana. And the idea of not disturbing natural ecosystems wherever possible is a real positive outcome. So rather than moving to a lower yielding banana that's got more disease resistance, if we can keep the same high yielding efficient one and fix the disease resistance issue, that's a much more preferable outcome, both agriculturally and environmentally. Wheat is another great example. So we brought wheat to Canada in the 1800s. It doesn't originate here, although we think of Canada as stereotypically about wheat. Wheat originated in the Middle East, and immediately we started breeding it to fit in our Canadian growing environment. But there's a lot of really cool gene editing possibilities coming for wheat. High fiber, so you can get more fiber in every loaf of bread. Low gluten to give more opportunities for those with celiac or gluten intolerances or market preferences toward low gluten. And also, again, back to that disease resistant piece. We're fighting a number of diseases in wheat like stem rust and mildew and things of those nature. And gene editing can really help address some of the challenges that plant breeders are facing in wheat. On the soybean side, there's a new gene edited variety of soybeans that resulted in a high oleic oil for cooking. This is on the market in the States right now. Less saturated fatty acids, less trans fats, higher healthy oleic acid. It's already available. It's one of the first commercialized examples of a product that's on the marketplace in the US. There's also another coming from Japan. There's a heart healthy tomato that they just started producing in Japan. So another really exciting example of what the technology can do there.